assign you to the breakout rooms. I let it automatically generate some. And I see, because not many people had arrived on time, that some of the rooms look a little small, but don't worry, I'm going to immediately change it so that you have more people in there. So I'm going to give you 10 minutes to make sure that we get through this. If you're in Group B, then you'll notice you have quite a few questions. There is a handout that can help you, and I can share it on the screen as you go through this, because that's all about the hormones and their regulation. And that handout summarizes it nicely. Or if you want, you can share your screen, and that way you can point at things instead of me trying to echo pointing at things with you. The rest of you have 2 or 3 questions, so I think 10 minutes should be enough. It's just Group B, but I think with the summary of that figure, I'm hoping in 10 minutes you can finish it, so we can get through all of the questions, since we have no way of spilling over into the next lecture. All right, so I'm going to open the rooms and assign the unassigned people at that point. Okay, see you in about 10 minutes. A lot of these questions, hopefully you studied for the lab test today, and we'll be able to go through them kind of quickly. And Group A are looking, they're kind of focused. The reason I grouped those 3 questions together is about some of the accessory organs, the gallbladder and the pancreas. So who is the spokesperson for Group A? I'm also going to share the screen in case you want to, if it works, in case you want to take some notes alongside of me.
I'll be doing number 16.
Okay, that sounds good.
So we said that bile flows out of the liver through the left and right hepatic ducts, which come together to form the common hepatic duct. And then the bile goes into the gallbladder via the cystic duct.
It's true, but it's backing up, right? So then it backs up. It backfills the gallbladder. Good.
And then the gallbladder stores the bile and concentrates it by absorbing water and some salts.
Because you can't move water without moving salts, so we better move both. Some salts and water. That's pretty messy, sorry. So the way it does it, as about the functional anatomy, is that it's lined by simple columnar epithelia, but they're doing absorption here rather than what you're used to along the alimentary canal, which is doing some sort of secretion. And it's doing that and so that you have a more concentrated bile that we'll use later when we talk about the digestion of fats. What about how does it contract? And tell me about that functional anatomy.
So ACH and CCK stimulates the gallbladder to contract, which causes the concentrated bile to travel down to the common bile duct and meet the sphincter of Oddi. And then the sphincter of Oddi opens via enteroendocrine hormones.
Great. And so another feature is it has 3 layers, third layer, which is an oblique one, right?
Yes.
So that when it contracts, it can collapse. And one other thing that was actually in the lab test, one other feature it has to help empty it is what? What does the lining look like?
Rugae?
Rugae. So when I'm asking about functional anatomy, then everything you said is right, but also focus in on the anatomy that aids in its ability to completely empty when it contracts. And then, as she said, then it goes by way of the cystic It goes out the cystic through the OD. In order to open OD, you also have to have CCK. And as far as I know, acetylcholine doesn't act on the sphincter of OD, but it does act on the third layer of the muscularis externae. So this acetylcholine rise occurs even as the stomach is receiving food. So what you'll find is the gallbladder will start some weak contractions, irrespective of the type of food that enters. So this is as soon as digestion starts. So we could say even during the cephalic and gastric phases in the stomach, and then it's increased as those phases proceed. But what triggers the CCK, which we're going to reiterate this a few times because we'll talk about it. What's causing the CCK to be delivered? It doesn't have to be you, Jessica, because I know this wasn't part of your question. Does anybody know what?
Proteins and fats present in the chyme.
Right. So now the food actually has to get to the duodenum. So you're going to get some much stronger contractions. These guys are going to be synergists with each other once the food has arrived in the duodenum, and it has fat and protein. So if you just somehow manage to eat just totally pure carbohydrates, the only way you're going to get CCK is by it being stimulated by the release of secretin. And then you won't get very much. So once again, kind of like in the kidney, try and keep in mind not just what is secreted, but how much. Because as you go forward in, say you ate something that had fat and protein in it, that's going to stimulate the ACH. And when the food arrives in the stomach, you'll start some contraction. Some bile will go down the cystic duct, but it can't exit until you have protein and fat, and CCK is going to be strongly released and therefore open that sphincter. So probably I should make a little more room on this thing since it's starting to look pretty messy. So those are good answers. I know it doesn't have very much in the book about the gallbladder under the little paragraph that covers the gallbladder. So that's why I'm sort of supplementing here as we go forward. All right, so in the pancreas now, there are exocrine and endocrine cells. One of the things I noticed is that many of you just labeled endocrine, exocrine without naming the cells, but of course, hopefully you knew what they were for the lab test, but also for the exam. So if we talk about the exocrine cells first, who's going to be doing that in Group A?
Oh, that'll be me.
Okay.
Okay, so the exocrine cells are the cinar and the ductal cells. And the cinar cells will secrete enzymatic secretions via CCK stimulation.
Okay.
And we could talk about the enzymes there in the next question.
Mm-hmm.
And then the ductal cells will secrete bicarbonate-rich secretions via secretin. And secretin is detected stimulated by pH receptors that detect low pH, I'm pretty sure.
Yeah, the pH in the duodenum. And we just said that CCK was stimulated by protein and fat in the duodenum. And then they also stimulate each other. So we could say plus secretin and here plus CCK. That way you're going to get at least some stimulation of the cinar cells, even if you didn't eat protein and fat, because the secretin is going to cause some secretion of enzymes by the cinar cells. The ductal cells are going to be most strongly stimulated by secretin, but again, and you will always get a drop in pH when something arrives from the stomach. So you'll always get secretin, but CCK will make a stronger release of secretin. Okay, those are the exocrine cells, and they make up the vast majority of the pancreas. What about those endocrine cells that are found in the islets? Is there someone else doing that?
It's still going to be me.
Okay.
So in the islet of Langerhans, you have the alpha and beta cells.
Okay.
And the alpha cells will secrete glucagon, and the beta cells will secrete insulin. And then glucagon is stimulated by low blood glucose, while insulin is stimulated by high blood glucose.
And that is true, but there will be other stimuli of insulin. So we're going to talk about a hormone shortly that will stimulate glucagon, GIP. So when we get to the hormones, that also can stimulate glucagon. And it itself is not stimulated by carbohydrate. It's actually stimulated best by fat. So these guys can be synergistic again with each other. So imagine you ate something that had both carbohydrates and fats, which we'll probably be doing a lot over the holidays. Then you're going to get the glucose absorbed from the breakdown of your carbohydrate into your blood will stimulate the beta cells. Meanwhile, the fat that's in your duodenum will stimulate the release of GIP, and that will also cause insulin. You'll get even more insulin release. Now, under some other conditions, insulin is also triggered that go back to 103A. I'm not testing you on that, but it's also stimulated if you are raising your blood glucose due to, say, growth hormone, or there are other hormones that will need the insulin. But we're focused right now on the stimuli which are associated with your digestion, which would be GIP and high blood glucose. Okay, great. So the next person, or the next question, the next lecture question, is about the enzymes released by the acinar. So are you doing that still, Noah, or is there someone?
Yeah, that's still going to be me.
Okay.
So for the acinar cells, the enzymes that it has are amylases, lipases, inactive proteases, and nucleases.
Okay.
And then we weren't too sure about why the proteases have to be inactive, but my guess is the same thing as pepsinogen, where you don't want it to damage anything as it's being secreted out.
Exactly. So because the way these get out of the acinar cell is that they have to go through a carrier. A carrier is made of protein. If it was an active protease, then it's just looking for pairs of amino acids for it to act on. And as they go through, it could in fact digest the carrier, which wouldn't be great. And it may digest other things along the lining, but you would argue the same for lipases there, because your membranes are made of phospholipids, but remember the phosphate group helps protect those. And everything else along the lining, carbohydrates, amylases, I suppose, could act on. But you do replace these cells. We didn't talk about stem cells, but you do replace these cells as well. So amylases are doing carbohydrates. Lipases are clearly doing fats. We'll come back to these later. And then there's a whole variety of proteases. There's actually, when it's saying NS here, these are fairly similar. Smaller numbers of these are needed, but you need a pretty wide variety of proteases because of the number of pairs of amino acids they're looking for. And for nucleases, all you need are ribonucleases and deoxyribonucleases. So one of the things with this activation, is that going to be covered later? I don't think it is. in any of the later questions. So when these are activated, they're activated two ways. And one way is they can be activated by the brush border enzymes. And that's required for the second one, especially if the enzyme is trypsin. So the enzyme used in the lab, trypsin, when it becomes trypsin, then trypsin can activate some of the other proteases. Not all of them, but it can definitely activate chymotrypsin, and it can activate more trypsin. So it's a little bit of a positive feedback. Activate other proteases. Sorry. So you're not entirely dependent on the brush border enzymes to do every single one of this wide variety of proteases, because trypsin is able to help out. And once it's activated by a brush border enzyme, it can then start activating some of the other proteases present. All right, great. Let's go on to group 2. And group 2, I'm going to stop sharing this, has a huge question with all of these different hormones. Let me share the other figure. And I think this figure that I had in my files is a little different than what you have in your Word document figure in Canvas. They're similar, but there's a few extra things on here. So I thought I'd put this one up because these are all hormones that are released from the enteroendocrine cells in the crypts of the duodenum. And then I stuck over here in a box, VIP. So let's start with relate the triggers that cause each of the hormones secreted by the duodenum with the effect of those hormones. So we've already touched on this, but it's probably worth hearing again. Is there a spokesperson that's going to be doing question 19?
That's going to be me, Kaylin.
Okay, Kaylin.
CCK is primarily stimulated by fats and protein in chyme, but secretin can also stimulate CCK in lesser amounts. Secretin is stimulated primarily by low pH and increased stretch of the duodenum, but CCK can also stimulate it. So therefore, more CCK will stimulate more secretin as well. And they both slow stomach activity, so that the duodenum has enough time to process the chyme. And then the CCK will increase pancreatic juices and contract the gallbladder and open the pancreatic sphincter. And then secretin will increase bicarbonate release from the ductal cells, and it'll increase bile production from the liver to replace the bile. And
Great. Okay, did you say something else? I'm sorry.
Oh, sorry. I said, increase bile production in the liver to replace the bile, and it'll also increase bile secretion with secretin.
Right, and this bicarb release is from those ductal cells we were talking about, right? So this is kind of vague. This is probably my old one, but I don't know. I don't know which one I made first. But the pancreatic juices are from the acinar, like Noah said, and then the bicarb is from the ductal cells. And I kind of made a general arrow backwards. And if I remember right, secretin targets the parietal cells more strongly to drop secretions. You can take a look. Yeah, that's what it says in that table 23.1. And the CCK goes back and with the stomach, if you look in that, it's more inhibiting its motility and its secretory activity. Plus, it goes back to, which I didn't put on here, goes back to the hypothalamus and tells your hunger center you don't feel as hungry. So that's going to suppress the stomach sort of indirectly by lowering the parasympathetic activation, suppressing that, increasing the sympathetic. long reflexes. So great. Okay, so where are you going to go on, Kaylin? And I interrupted you with the CCK and secretin story.
And GIP is stimulated by fatty chyme, and it stimulates more insulin release to take up that glucose. And then intestinal gastrin is secreted initially in the intestinal phase by decreased pH and increased stretch, and it'll increase the stomach motility and secretions really briefly.
Great. So actually, this is the first of them that are released, but it ends pretty quickly. And you'll see in the book, GIP used to be called gastric inhibitory peptide. So it does go back and inhibit the stomach, but it's a very weak effect of it. So they had to, as they learn more, they realize its real target are the beta cells in the pancreas. And they actually had to change its name, but they wanted to keep GIP the same. So they had to be kind of creative in doing that name change and called it glucose-dependent insulinotropic peptide. So it's kept the GIP, but now the real name is telling you that it's targeting the increase in insulin by the beta cells. Okay, so the only addition I think I had on here is motilin, and motilin is also released from those enteroendocrine cells about every 1 to 2 hours on a kind of circadian rhythm. You see a greater motility along your entire GI tract. So once we get later into the large intestine and the rest of the small intestine, it's acting there as well. And the stomach too backwards. It's just generally increasing motility. But with it using short reflexes, once food is there, that tends to go up as well. So when it's saying neural circadian, that's supposed to say circadian, but it got split, rhythms, the circadian rhythms act regardless of whether you're eating. The neural ones are part of the short loop that encourages the movement of the whole alimentary canal. So I tried to include most of the hormones that we are talking about, but one is missing, and that's somatostatin from this figure. I think it's missing from your figure as well. So let me maybe switch out since I can't write on this one. And have to wake this thing up. Hold on. Oh, come on. Try again. Okay. So let's share this again because we'll go forward with it. And what I'm doing is basically going through that table and trying to put each of those things in the table into context. So the last hormone, the ones that is somatostatin. This is a funny thing because it's released both neurally and it's released by your duodenal enteroendocrine cells. So the book is mostly focused on the release by the enteroendocrine cells, and this is actually triggered by the food in the stomach. So food in your stomach will, through sort of a neural short reflex, cause not only a neural release of somatostatin, but also the duodenum to start releasing somatostatin. And what that's going to do is suppress your stomach through, again, decreasing its motility. And it's going to often do it, by way of increasing norepinephrine, epinephrine. I can't write on this thing today. Epinephrine's effects. So this one's a little more complicated. It actually also goes and interacts with the endocrine part of your pancreas. But we'll just go with what is in the table on that, where it says it inhibits gastric secretion. So the stomach is inhibited, its secretion is inhibited. In the pancreas, it tends to decrease secretion as well, which means mostly the exocrine cells. And it inhibits contraction of the gallbladder and the small intestine. It's basically slowing your digestion so that things stay in the stomach a bit longer, and then, you know, motility. These things are inattention is what I guess I'm trying to say. So as food arrives in the stomach, it tends to stimulate this great motility and secretion, and somatostatin is working through short and long reflexes to kind of slow things a bit more and keep things in the stomach a bit longer. So that brings us to 20, where I already introduced a couple of the neurotransmitters that are involved in slowing it. So in a way, somatostatin is one of them. I've already kind of done part of, because it is released both neurally and, he's loose, Kyle. Oh, he's not. Okay, sorry, my dog I thought was loose. Okay. It's released both neurally and indirectly. Norepinephrine and epinephrine is involved in this. But what is the other one? Who is doing 20?
I'm going to answer 20.
Okay.
So I named vasoactive intestinal peptide, or VIP. That can induce smooth muscle relaxation in the gallbladder and the stomach. It can also stimulate the secretion of water into the pancreatic juices and bile, and also causes the inhibition of gastric acid secretion and the absorption from the intestinal lumen. And the other one I said was norepinephrine. That causes the liver to increase the breakdown of glycogen, protein, and fat to increase blood glucose, and can also decrease digestive and salivary secretions.
Okay, the other thing in the table is this increases the secretion of bicarbonate. So, secretions. So remember, we had lots of sources of bicarbonate. We have those duodenal glands, we have the bile itself, and we had the ductal cells. And this tends to increase that. And as he said, it does relax smooth muscle and increases some secretions and inhibits some others like the acid. But one of the main things it does that he left out, I'm just going to put this, is a little bit of an asterisk, since it's sort of hidden in that list on the table as well, is it causes vasodilation in your gut. So why you want that is you want more blood to flood into the capillary beds around your duodenum and your liver and your stomach, for that matter, all the other organs as well. But think of it, you're going to be picking up all these nutrients, so you need to send more blood there. Plus, everybody's going to become more active. They're going to need more oxygen delivery, etc. So this is probably one of the more unique things, since a lot of the rest of this could be said about other hormones. It's kind of redundant with other hormones. But here we have with other hormones and certain neurotransmitters. But here we have the neurotransmitter that's causing some major vasodilation of the arteries going to your GI tract. Okay, good. All right. So how about 21? We kind of did 21, but why not hear it again, since this asked you to compare and contrast secretin and CCK.
Okay, I'll be doing 21.
Okay, maybe I can go back to here.
So as chyme enters the duodenum, it causes the release of CCK if there's a lot of fat and protein present, but some will be stimulated by CCK by secretin, and secretin will also be secreted regardless, since there's always low pH and stretch with chyme, and CCK can also stimulate secretin. CCK also induces acinar cells to secrete enzymatic rich pancreatic juice, and secretin induces ductal cells to secrete bicarbonate-rich secretions, and secretin will also stimulate bile secretin, secretion from the liver and increase bile production to replenish bile. And CCK via the bloodstream can cause gallbladder to contract and the hepatopancreatic sphincter to relax so that bile enters the duodenum.
Great. So the gallbladder and the sphincter, right?
Mm-hmm.
OD contract. With CK. So there's a nice figure in the book, where they use the red and the yellow dots to kind of show all the targets. So these two are talking to each other, or at least through their enteroendocrine cells, complementing each other. And then at targets, they are both targeting the pancreas, but looking for different cells. And when the secretion, I wrote, should have been secretin, not secretion. Secretion is going to increase bile production so that as you empty your gallbladder, you're now preparing the liver to refill it. The CCK is causing the gallbladder to empty by contracting it and then opening that sphincter. So the bile will then leave, and meanwhile, the secretin is telling the liver, you got to make some more bile. So you can then refill the gallbladder after all of this is over. Okay, and I did also mention earlier that CCK goes back to the satiety center in the hypothalamus. So then you don't feel quite so hungry anymore. This is all before lunch. It's kind of hard to hear about, huh? Okay, so the last one in this kind of long group is GIP, which we did talk about already. Was there someone I've gotten ahead of there and talked in their place for number 22? Is there someone who's going to add anything?
Yeah, that's me.
Okay.
So yeah, we did talk about it. So it's basically fat are the best stimulus for GIP. And it will react on insulin release for B cells. And it will also have to do with satiety at the hypothalamus as well.
Okay, so in the way, this doesn't make, in some ways, a little unexpected maybe, that fat would then cause you to release more insulin. So, but remember, insulin is not just targeting your liver and telling it to take, and store glucose. It's also targeting all the cells of your body and increasing metabolic rate and the kinds of metabolism it can do, like it supports protein synthesis. So, in a way, it does make sense because you're now going to be giving a permission slip to cells to become more metabolically active and start doing a lot more anabolism by the cell, and therefore they may need that fat that comes in and it's being absorbed in your duodenum may be able to supply energy for doing those processes. Okay, let's shift gears here. We're going to group C, and they're looking at some of the other organs. So who is doing 23 and looking at the large intestine, cecum, and the appendix? Item 1. Who's in group C? Adrian and Ileana, I have. One of you guys talking?
Yeah, I can do question 23. So
Okay.
the function of the large intestine is to absorb some of the water. The bacteria that resides in the large intestine also generates vitamin K and some types of vitamin B, and it also expels feces. And food clumps down in the cecum, and microorganisms digest it. And you can also educate the microorganisms through the appendix. The appendix hangs off the cecum and contains crypts, which is part of malt, because the cecum is where food gathers and comes out of the ileum. Yeah.
I got my notes switched here. Okay. Great. Perfect. There's a lot of interest in what's going on in your large intestine, and especially in the microbes that are in your large intestine and the way they interact with both what you're eating, but also with your sense of hunger, satiety, body weight regulation. So the types of microbes that you cultivate in your large intestine seem to have much wider range physiological effects than just being a manufacturing site for some of the vitamins. So some of the B vitamins are made in your large intestine by bacteria and help you. Plus, they do compete with other bacteria, so they have immune functions. All right, there isn't, again, a lot in the book on that one, on those others, so I just supplemented it a little bit, but all right, so let's go on to these sphincters, the anal sphincter, and it asks you to compare and contrast the anal sphincter with the sphincters that control micturition. So what's similar about both the urinary and the anal sphincters?
So both open during micturition or defecation, and in both the external sphincter is innervated by the somatic nervous system, and it's made of
external.
Excuse me.
External somatic, right?
Yeah. And it's made of skeletal muscle, while the internal sphincter is innervated by both the parasympathetic and sympathetic nervous system, and is made of visceral smooth muscle. And sympathetic in both is active at rest to hold either urine or feces. And I didn't.
So this guy would be an inhibition, right? And this guy would be excitation. Mm-hmm. And at rest, it's excited. And this is inhibited at rest.
My group couldn't really find a difference besides what they let out, or the differences.
Okay, there's so many similarities, and I mentioned this earlier. You're right. There's a lot of similarities. So what triggers these things is a stretch reflex, right?
Yeah.
Stretch receptors that are in the wall. Now, with the only one I mentioned as far as a difference when we were doing the lab was with the bladder, you have a certain amount of stretch. Every 200 mLs will keep telling you, you've got to go. And at some point, it's going to override. This stretch receptors go to both a local reflex, and this is true for both of them, and it goes up to your CNS in case of in the case of micturition, it's going to the higher centers that then regulate pontine nuclei. In the case of defecation, they didn't name in the book, it's what part of the brain, I actually don't know. But again, it goes to the higher centers that then go to nuclei that then can suppress your sympathetic and relax your somatic. So this is going to end up suppressing if you're going to actually defecate or urinate. And somatic, if you want to have it happen. If you don't want to have it happen, if you want to hold, so when you expel things, then you're going to suppress further your reflex. So maybe I'll put that one in a different color. So the reflex on its own is just going to run, maybe I'll stick with that. If it's just the reflex on its own, like in an infant, then the reflex alone will lead to both expelling. Meaning that reflex will suppress, there isn't any somatic in an infant to concern about, and your sympathetic isn't well innervated yet, so it's expelling it via parasympathetic local reflex. This wasn't clear to everybody in the exam 3, that you have these stretch receptors that aren't just going to the brain, because after all, in an infant, Those pathways aren't there yet. Even in an adult, you have this local reflex that will do its own thing unless the brain is saying, nope, you got to hold. The default is holding. So in order to say, if you're trying to expel in both the cases, then you've got to have your CNS, and that's what I was switching it to. If you want to hold, then the CNS has to go and suppress that parasympathetic. And then it's going to also increase your somatic and your sympathetic. So get this down if you didn't get it all in the last exam. So what are the differences? Well, probably the main difference is that there isn't this 200 mLs amount. You reach a certain degree of stretch and you really need to go to the bathroom to defecate. But if you decide to hold, you can really hold a lot more feces, assuming you don't have some other issue like, you're ill and you're having diarrhea that's causing, a great amount of motility in your GI tract. But generally speaking, you can hold a lot more feces and a lot longer, and there isn't this sort of stepwise increase in excitation. Okay, so let's go on to, well, we just did that one, 25. Is there anything that's about holding? Is there anything that you want to add to this? Whoever had 25?
I think it was mostly covered already. We didn't have anything else to add to holding.
All right, so let's move on to group D, which is again a shift in topics. And they're going to be talking about where and how things are digested, which we mimicked by our little lab videos. So each of the macromolecules, so carbohydrates, proteins, lipids, and nucleic acids, right? Who is doing 26?
It's me. For sugar, in the mouse use the amylase, and in the duodenum use the pancreatic amylase to digest the sugar.
And what are the final products?
The final product is for them is the disaccharides.
And even larger too, right? Oligio, which would be 8.
Dextrins.
We good? You got cut off there. So these are still not able to be absorbed, is the point. They're large enough, even if they're a disaccharide, you can't absorb that. Oligio have I think it's eight from the prefix, sounds like it is, 8 sugars bound to each other. So amylases are mostly being done in I'm going to put a star next to the pancreas because the pancreatic ones in the duodenum, because you don't keep food in your mouth long enough to really digest them. And if you wanted to try a little experiment that I know they sometimes used to do in elementary schools, you go buy saltines with no salt on them. Sounds like a contradiction in terms, but they have unsalted crackers that are basically pure starch. Maybe there's a little fat in there too. I don't know, but there's no salt in there. And if you stick that in your mouth and you leave it long enough, it will start to taste sweet. So So the reason you don't want salt is it kind of hides that sweet taste that you might detect, but you have to hold it in your mouth a really long time before you can taste any of the sweet taste of the disaccharids that are being made from the starch. Okay, how about proteins?
Proteins in the stomach, pepsin.
And what is the product there?
Is I think is the polypeptide.
And they're large polypeptides, right? So you don't get very far. You denature it with the HCl, and then the enzyme is pepsin that's going to be acting on it. But you haven't gotten more than unwinding it and having these huge, probably still maybe 100 or so amino acid chains, certainly nothing you're going to be able to absorb. And then what do you do, John?
In the duodenum, there are some enzyme like trypsin, chimer, trypsin, and carbon peptides.
Okay, and what are they? Many I'll put. And what is their product?
Product, I think, is the more smaller.
Yeah, smaller high and try. Okay. Peptides. Still can't absorb these guys, or at least not many. Sometimes you do absorb some that are brought in by endocytosis. So what do you need lastly to finalize this?
Amylopeptides on the brushboard.
Right. The brush border of the duodenum. And that'll make, and there's, again, many of those. You can hopefully have learned a few names. And that's going to make amino acids. That now you can bring those in. Okay, how about lipids? Are you doing all of this? This is a lot. So
lipids in the mouth. In the mouth, there are little lipase, and the stomach are a little.
And we don't really know that there's much products made from this. So I'm just going to leave that a question mark, but I suppose there could be some monoglycerides made, but in fatty acids, but not clear how much that. So where is most of this digestion?
This is pancreatic lipids.
Plus the bile.
Bile, yeah.
And now you're going to get fatty acids and monoglycerides. So I hope these words are making sense to all of you. A glyceride is because there's a glycerol. Triglycerides have 3 fatty acids. Mono have 1 with the glycerol. And di have 2 fatty acids in a glycerol. These are still really pretty big molecules, not the fatty acids as much, but the mono and diglycerides. But not to worry, they're fat soluble. So when we go to absorb them, even though they're big, we're going to be able to do that. Okay, how about finally the nucleic acids?
In the duodenum, use the nucleus by the pancreas.
Yes, and that makes, and I'm running out of nucleases, and that makes nucleotides, but we have to still break that down further. And who does that?
On the brush border, use the brush border cells to break the nucleotides to phosphate on nucleosides.
Right, phosphatase.
And
all right, okay. And nucleosidases.
Then,
And what does that mean?
nuclear size is break down to the to the 5C sugar or and N base.
And a phosphate. Yeah, nucleosides, you're right. And nitrogenous base. And then you're going to also have the phosphate because of the phosphatases. So that's what's absorbed, are those molecules. Okay, 5-carbon sugar because ribose and deoxyribose have 5 carbons, but they're still a simple sugar. The nitrogenous bases are a bit bigger, but they apparently can be brought in by a carrier. All right, so that was quite the mouthful. Thank you. Let's see, where are we on 27? And I think we did some of this already, but since it was a question on the lab test, it asks you to define emulsification. Who's doing that for us? Okay, Stephanie. Okay, suspension, suspends, that insoluble. Suspend, within a fat insoluble. Maybe I'll start over again. I was trying to keep up with you here. So you're going to suspend something that's fat soluble in the digestive juices, right? Maybe I'll just suspend fat droplets in water. Okay, and you do this by the fact that these are amphipathic, which is another term that it was supposed to be. So if that's the fat, what's an amphipathic molecule doing? So it's putting its phobic part near the fat, and it's putting its philic part out to interact with, perhaps I should have made the water blue, but with the water out here. And so these guys get surrounded by like a shell of bile. Now the droplets are pretty big, but they're still smaller than they would be otherwise, and that allows your lipases to access more surface area. So you've increased the surface area for those lipases to act on, which means as they act, the droplets are going to get smaller and smaller and smaller. And as they get smaller and smaller and smaller, eventually still surrounded by some sort of amphipathic molecule. What do you call them when they're really small? Micelles. And by the time they're a micelle, what that is surrounding are the monoglycerides and diglycerides, as well as things like cholesterol and the fatty acids, and glycerol probably isn't necessarily in there because that's water soluble. So these micelles can be surrounded by bile, but another molecule, which is that in your list? Yes. What's the other molecule that could be surrounding them? Yeah, lecithin. It's a hard one to say. It's actually part of the bile salts. It's one of those, why they don't just call it bile, they call it bile salts because they're different components. And by the time they're little tiny micelles, that bile and bile salts are, one of them could be lecithin. The book mentions it, so I put it in there. It's actually a phospholipid. This is a phospholipid. And that, particularly the lecithin, is going to help that little tiny micelle shuttle up to the surface of the absorptive cell, when we talk about that soon, and fuse that phospholipid with the phospholipid of the lipid bilayer membrane and help the fat get through that layer by simple diffusion. Okay, perfect. All right, going to question 30. I know these are out of sequence, but it just seemed to make sense. We're going to absorb things now. Who's doing 30?
It's me.
Okay.
For the sugar,
Yours. Mm-hmm.
for the sugar, the glucose and lactose are absorbed with co-transport with sodium.
Okay. So if this is the absorptive cell, the blood's over here.
Yeah.
These are going to go in with sodium, and what kind of active transport is it?
Secondary activity.
Right. So this is a carrier here, secondary active. Now, once it gets inside, how does it exit?
Can you see that?
Going to use a carrier over here.
I'll use the facilitated diffusion.
Right. Facilitated diffusion. Now remember, if it's glucose, it's going to become glucose 6-phosphate. So we're going to need that glucose 6-phosphatase to convert it back to glucose and ship it out. But other than that, the other sugars are going to mostly come in by secondary active transport with sodium. So there's one exception.
Fructose.
Fructose, right.
Use the facilitated diffusion.
And it apparently uses it on both sides. I'm not sure how that works, but apparently it uses it on both sides, maybe because our body doesn't really make fructose except metabolically. So maybe it's always low in our blood, and so it's pulled over into our blood by facilitated diffusion. So fructose does tend to raise your energy pretty quickly because it's moving just through facilitated diffusion. In order to have this sodium secondary active transporter, what do we have to have on the basolateral membrane here that is not present on the
Sodium and the potassium pump.
right? The sodium potassium pump. So really, except for the fructose story, what we're talking about, at least in the upper part here, I'll maybe put it in parentheses, is like the same as the PCT. You're just basically seeing a PCT type cell as far as these guys. It's just that this is your food instead of your filtrate. But same idea there. It's fructose gets that exception. Okay, great. Thank you, Jen. So how about the other ones? What about amino acids?
Amino acid like sugars.
Oops.
And
So they're like sugars?
yeah, use the co-transport with sodium. In and out, use the facilitated diffusion.
Right. So again, just like the PCT here, but there is an exception that's a little different than, of course, there always has to be one. Some amino acids don't use sodium. What do they use?
Use the hydrogen.
Right. So hydrogen, because of that low pH that's out there from the stomach's acidity, is going to be high out here just because of the secretions of the stomach. So the duodenum can exploit that. Now, I don't know how it doesn't cause pH change in here. There must be some sort of way in which that is either buffered or transported, out back into the blood, or I'm not sure why, but certain amino acids do take the H plus It's just kind of an elegant way to exploit the fact that that's always going to be at a lower pH out here to use it as these are all considered secondary active transport. And this goes out by facilitated diffusion, doesn't exit any differently than amino acids coming that way. Okay, sugars and amino acids, what takes them out? So that finishes up group D. How are we doing on time? We're doing okay. So let's go on to the last group, group E, which is a story about fats. So we shuttled those fats up to the membrane, and these little micelles, little teeny droplets, have mono and diglycerides in them. There's probably fatty acids in there. And there could be cholesterol in there as well. And then this asks, how are they absorbed? So now we're going to have that same absorptive cell. And this is how they're entering. And then how they exit. And then where do they go? So who's doing 28, which is another kind of long answer?
That would be me, Jeanette.
Okay.
So we refer to a figure in the book, and fatty acids and monoglycerides enter the intestinal cells via simple diffusion. The fatty acids and monoglycerides are recombined to form triglycerides and then combined with other lipids and proteins within the cells. The resulting chylomicrons they then leave by exocytosis. The chylomicrons enter the lacteals of the villi and are transported to the systematic circulation via the lymph in the thoracic duct. Some short-chain fatty acids are absorbed, move into the capillary blood in the villi by diffusion, and are transported to the liver via the hepatic portal vein.
Okay, and how do you get things to move through this? Sorry. Go away. Go away. Siri, go away. I can't even get Siri to listen to me today. I'll just turn it off. Okay. So how do they get out of that lacteal and get moved down through the villi and then eventually, moved through the gut?
It drains via thoracic duct.
Well, there's a term I was looking for, milking.
Oh, okay.
And that milking is done by the muscularis mucosal, smooth muscle. Remember that we had to have that muscularis mucosa before in order to cause churning, which it does, but part of that churning movement makes the villi go up and down, and this is kind of like a milking movement on those lacteals to draw the fluid through the lacteal to the larger lymphatic vessels, and then undoubtedly the gross movements of the muscularis externae continue the movement of that to the lacteal. So this is good. This is great. Now I'm not sure why in the book, but they kind of separated that. This chylomicron we'll talk about a bit more in a minute, but that some of these guys don't do that, but they exit and go as mono and diglycerides directly to the blood. They don't really go into that a whole lot. So some of the mono and diglycerides will go directly into the blood, but the vast majority are going to be packaged as these chylomicrons. Now, why I wanted to bring it up is not only because it's accurate, but also remember that this blood is going to be triggering things like the release of GIP, and also it's going to be, through the fats, it's going to be transported to the liver directly without affecting all the other steps that we're going to go forward with. So let's be, they do talk about this a little bit in the book, that there is some of the mono and diglycerides that don't have to get packaged into these chylomicrons. Now, they don't really talk about how they get in and out of the cell then, but they probably get in the same way, which would be simple diffusion. But once they're inside the cell, the reason you have to make a chylomicron is the cell is full of water, and this fat would separate into a separate little globule. So how this happens, they don't really explain, but we want to acknowledge that it does. All right, so let's talk about these chylomicrons. What are they? There is a really good figure. I'll give you the figure number in a second. It's in chapter 24, and it's 2424 that you want to look at. So let's see what I've gotten out of my questions here.
It was 29. I think.
29 now, right? Compare and contrast these various things, which that figure does really nicely. Who's doing 29?
I was just going to do 29 as well.
Okay, Jeanette, thanks.
So the chylomicron carries absorbed lipids from the intestines to the liver. And then the VLDL, which is very low density lipoproteins, carries triglycerides from the liver, mostly to adipose tissue.
All right, slow down a second. Before it gets to the liver, remember it's in the lymph. Well, maybe I should do it as well. I should slow down as well. How does it get from the lymph into the blood? This is good for review too. Where does the lymph dump into the blood? Remember this from the
Thoracic duct?
Right, the thoracic duct. So remember all that cisternae chylae, where it all kind of coalesces from all your gut and your legs. All that lymph then travels up that thoracic duct. Oops, dang. And it is dumped into your blood. So once it's in the blood, some of the cells that line the blood vessels will act on these chylomicrons. So eventually most of them go to the liver, but some are actually acted on by the endothelia of the lining of the blood vessels. And those, if you want to go back again, good for review, those are really busy cells. They're doing all kinds of things, preventing blood clotting, and once blood needs to clot, they help the blood clot. There's all kinds of things these endothelial cells are busy doing, but one of them is they do convert some of these chylomicrons. So we'll just make two arrows here. We'll put endothelia conversion. To allow some fat. But as you say, the most of it is going as a chylomicron to the liver, and adipose tissue can also deal with this. But what's it doing in the liver then, since I interrupted you? What happens in the liver? Are you still there, Jeanette, or are you muted?
Oh, sorry. Oh, okay. The liver. I'm actually sorry. I'm a little lost.
Okay. I kind of threw you off. You were talking away, and then I sort of interrupted you.
Oh, I'm just describing the chylomicrons, correct?
Right.
Oh, okay. So yeah, the chylomicrons carries absorbed lipids from the intestine to the liver. Then the very low density lipoproteins will carry triglycerides from the liver, mostly to adipose tissue. Then the low density lipoproteins, so just the LDLs, carries cholesterol from the liver to tissues.
Okay. So
And
the VLDLs that you're talking about, they're carrying it to the from the liver, but mostly to adipose tissues, as you mentioned. And then what are the adipose tissues sometimes called upon to do? They make them into what? I probably interrupted you again in your story.
For LDLs, they carry cholesterol from the liver to the tissues. Yeah.
Yeah, so LDLs from the adipose tissues to the body cells. And then finally, the other sort of side story with the liver is, or with adipose is, maybe I'll make it a different color, is it can, adipose can make them into HDLs. What are HDLs doing?
They carry cholesterol from the tissues to the liver.
Right, especially adipose, but it could be other tissues to the liver. So this figure shows you what the contents of these things are. So I would at least have some sense, not the numbers, but if you're looking at a chylomicron, the vast majority of the chylomicron, where are they here? I think they get, I won't give a number, but I'm going to say, greater than 80% of this is triglycerides, what's inside of that. This is what you absorbed from your diet, and fairly low percentage, they give 2 to 7, is cholesterol. So this is fairly low in cholesterol, high in triglycerides. So on a blood draw, these things, chylomicrons, plus the stuff that you absorb directly, the stuff over here that's in green, this and this in your blood are often labeled triglycerides in your blood draw form. If you ever have your blood, look at your blood tests, it will be looking at how much triglycerides. And so they're really looking at what did you just absorb largely in your food. So if most of the time people do blood tests when they've not eaten overnight, but even then, more recent, since digestion takes many hours, that could be that you had a high fat diet the night before, and you may see triglycerides that are elevated. So triglycerides in a blood draw are going to fluctuate with what you've recently eaten. And that's one reason why they want you to wait and not have your blood test done immediately after a meal. So they have an older sense of what's going on. So then if you go down and you look at what those are converted into, these very low density lipoproteins, those have increased. They've really 2 times as much cholesterol as the previous form, and substantially less triglycerides. Now, there's other stuff that they talk about, protein and phospholipids, which we won't really worry about. But these guys are then being taken by the liver to send this to your fat cells. Now, in your fat cells, they're going to be converted into some sort of storage form of fat. So they don't go right from VLDLs to LDLs. They're stored here in some sort of, in vesicles, in some kind of format. Now, the reason you're storing fat is obviously for energy, but also for cholesterol. You do store cholesterol so that when you need it, your cells are needing to grow, they will, you will start to break down your fat. Remember, these fat cells are making a hormone as well. Maybe I'll make it a different color. They're making leptin in proportion to the amount of fat. And that could influence your hunger and satiety center. So if that leptin store drops below whatever is set point for that cell, or that fat drops below whatever is the set point for that cell, it'll start releasing leptin, which will then make you feel hungry. And then let's say your cell does need some cholesterol, needs some fat. It's going to undergo mitosis. It will sprout receptors for these LDLs. when it needs to take fat out of the blood. So you're all often in a constant conversation. Why I'm going into this a little more is you probably will hear quite a bit more about this with blood draws. They don't look at VLDLs and blood draws, but they do look at LDLs and HDLs. And they often call this the bad. And it's not inherently bad. I mean, it's just a way, it's just a shuttling system for how you move fats through the blood for different destinations. So by having a specific receptor for LDLs and that don't take up chylomicrons or VLDLs, then you know where the fat is going. The problem is apparently inflammation will cause these LDLs to form plaque. And the LDLs are 3 times as much. So we'll go 3 times as much cholesterol in those than in their previous iteration. So as you're going, you're getting more and more and more cholesterol. And so these are cholesterol-rich. Inflammation tends to make these things adhere to the walls of vessels and cause arteriosclerosis. So they're not inherently in themselves bad. It's when you have some kind of chronic inflammation condition, at least that's what's believed now, that plaque forms. So you do try and keep your LDL levels low, only because that is telling you that, you know, there's a chance you'll get more plaque deposited if you are having this inflammation. So I'm not sure what happens if you go to suddenly lose weight and you download a lot of these LDLs to body cells. You know, maybe there is more of a chance of getting plaque in those conditions. So the last one that I put over here are the HDLs. Maybe I'll change it back to that color. And these are kind of out of the loop. HDLs are the lowest of all of the density of the, I'm sorry, they're not density. They're the highest of all the density. They do have quite a bit of cholesterol. Let me see. They have about the same, a little between the VLDLs and the LDLs. So there's less cholesterol than an LDL, but that's not the reason why they are considered the quote unquote good. You want your HDLs to be high and your LDLs to be relatively low in a blood draw. It's more to do with most of the HDLs, not all of them, but most of them are traveling from adipose and other body cells to bring cholesterol to the liver, where it will be converted into bile. So that cholesterol that's being made is being transported is eventually going to end up in bile and therefore be, well, excreted is sort of true. You are going to lose some of it in your feces. You do recycle your bile, but it's not 100%. So you're constantly making new bile, and you need that cholesterol in order to do that. So one reason why HDLs are considered the good guys is because therefore you're going to be lowering the amount of cholesterol that is in your body cells, but also they don't seem to be correlated with the formation of plaque. So even though they're carrying cholesterol and a fair amount of it, they don't seem to respond the same way and form plaque. Now, these are big groups of molecules, so there are some not so great HDLs and some, better LDLs. So it's not as black and white as this is portraying, but certainly on a blood test, that's the kind of thing they're looking at. Okay, phew. So, sorry, that was kind of messy, but that finishes up, I believe, right? Everything on that 30. Am I done there, Jeanette? Are we done?
Yeah, that was the last question.
129, right. That was the last question. So we're about right on time, actually. And so I just give you a few reminders. Then I did move the due dates. Let me stop sharing this. This messy thing. I did move some of the due dates. So the lecture questions, I think I made those due, well, you can double check, but it's on the 15th. And I believe they are due the 5 p.m. That's the part I'm not sure if I'm remembering right. The 15th is Tuesday. So Tuesday, and I believe it said 5 p.m., turn in the lecture questions, but I would try and write as many of those and as many of the essays as possible before the review. And the review is the normal lecture hour, 9:20 to 5 of 11. And what I did, because I have meetings that afternoon, I did move my office hour. So I have an 8 a.m. office hour. So if you have something you need to talk to me about before the review, or you think it's a question only would serve you and nobody else, then you're welcome to come at 8 a.m., 8 to 9 like usual. But instead of the afternoon office hour at 2, I made an office hour that I think I gave myself 15 minutes after our review, and then have an office hour for half an hour. So I believe it's at 11:15 or something like that. I set it up. So those are mostly if you have some personal questions or some other issue. We should, with an hour and a half, have time to plan to go through an old exam. We can go over the essays if you'd like for the last midterm. And for the comprehensive, what I'd suggest is pulling out your old midterms this semester, old essay, piles of essays that you've taken, and go looking through those to see if there's any questions you want to review. I don't know that there's any point in looking at an old comprehensive, but we could. I posted it, since I doubt that I'm going to make it look like when we had it in person. I think last semester what I did is I had only essay type formats. And as I told Noah, many times what I do is I pick a theme, and then I ask, you have some choice of how you want to address the question. So I think the example I gave him is something like, use 2 body systems to discuss the role of calcium in the physiology of that system, something like that. So it's not, right out of any one essay or any one earlier midterm, but it often will touch on more than one body system. Sometimes I've done case studies which do the same thing. I'll give you, this patient has, hypercalcemia, tell me, about how it's going to affect their heart and their digestive tract or something, something like that. I'm just doing this off the top of my head. Or high blood osmolarity, what's going to happen, something on that order. So I think it's a little harder to use the old exams, is the point I'm trying to make, to study for the comprehensive, the old exam from when we were in person, the one that's posted. But I'm happy to go through it if you'd like to, just to kind of remind you of what material, where we've been. What I don't think is very useful is to just try and reread the book. You'll just go to sleep because you've read it once already, hopefully, and it's a lot. So you'll spend a lot of time, and I imagine your brain will drift off and won't be focused on it. I think your studying for the comprehensive should be a very focused study and to things you don't remember. And the best way to know you didn't remember or you never learned them is to kind of look at the old midterms this semester and try and see what you missed and whether things that you wrote back then and your essays that you prepared still are familiar to you. It also will mean there's not going to be a lot of super small detail questions, because there are going to be these, shorter answer questions. Okay, so any questions before I let you go? Okay, so I didn't take roll, but we have 18 people here. I don't know if they're still here. I will be here at 1 o'clock. So if you have questions in 10 minutes or 15 minutes, I'll be here. If we need to, I'll go through the lecture, but I'm mostly it seems like the people everybody's still, the people who couldn't come to this lecture are either no longer in the course or they've found a way of coming to this lecture, this lab section, and hear what goes on in the lectures when I have them. But please, you're welcome to come if you'd like. I'll be around, and I may cancel my office hours if that's the case, if I'm not having to, go through the whole lecture, the late afternoon office hour, just because the next hour and a half will be basically an office hour otherwise. Okay. All right. So happy studying, and I will see. Don't forget to turn in by the way, don't forget to turn in your lab stuff from the digestion processes lab. I saw this morning when I looked, not very many people had yet turned it in. And probably could have helped you on the lab test, the last question. So do turn that in today by 5. So, okay, I think that's everything. Is there any questions? Thank you, Kaylin. Okay. All right, then I will let you go and see you on Tuesday. Bye.
Bye.